Okay, it really is an honor to be part of this session. David keeps thanking us for coming, but I'm really honored to be part of it. And I want to tell you about some work we've been doing with Droplet Microfluidics. And before I get into the science part of the talk, there's a couple of things I want to do. First, I want to acknowledge the students that are listed here who've done the work that I'm going to describe, and I'll try to mention their names as I go along. I also want to acknowledge our collaborators. David Lombard is a professor at Michigan, and who's helping us with some of the applications, and Gary and Mike helped us with some of the initial experiments that we have done. And our work would really not be possible without having these great collaborators. The second thing I want to do is congratulate David for this award, which is so well deserved. I mean, one of the bad things about these award sessions is you have to speak after the awardee, and it's a little bit intimidating. Actually, it's an awe-inspiring talk, I thought. One thing that, as he mentioned when he spoke, is that he is an alum of the University of Michigan, and so as now a professor at the University of Michigan, we're very proud to claim these alums that have gone on to be so successful. I guess we can't promise every student who comes into Michigan they'll be as successful as Dave, but we certainly claim to give them the best chance, right? And I like this little thing up here. It says, "Always a Wolverine." This is put out by the Alumni Association, and I'm quite sure that the Alumni Association never lets David forgets that he is an alumni of the University of Michigan. But seriously, one thing I want to point out about David that many people may not be aware, I mean, many of you are aware of his great scientific accomplishments, but he really gives back to the university in a lot of different ways. He comes back, provides a lot of advice to the college. He was on our external review for the chemistry department, put a lot of effort into that. So he really maintains himself as a member of the community and helps with those things a lot. So for that, we're also very appreciative. So what I wanted to talk about today, as I said, is some work we've been doing in the area of droplet microfluidics. And this is an area that's really been exploding over the last few years, both in engineering and in chemistry. And I think in terms of chemical analysis, it's something that offers a lot of opportunities for automation, miniaturization, and throughput. And I'll try to hopefully show you that today in my talk. So the concept here is we're typically working with two-phase systems. Often we think about aqueous-type samples that are in some sort of oil. We typically use fluorinated oils because they really give you good immiscibility. And aqueous samples can be injected into oil to form little tiny droplets. This is a femtoliter volume droplet that's from Daniel Chu's work. They can also be made a little bit larger, so they actually span the walls of a channel, and those are called plugs. But either way, they're essentially droplets within oil. And what's interesting about this, at least the first stage to us, is that these little droplets can act as your reaction vessels or sample vials that you can do chemistry in. And they can be obviously much smaller than what you can think about making in other ways. And those of you who've been around a while might recognize this, especially this plug-type flow, essentially a miniaturized form of something that is sometimes called continuous flow analysis or segmented flow analysis. And I think one reason this has been revitalized a little bit is some of the things that have happened in microfluidics allow us to manipulate these samples. And so I want to show you that first. This first movie is an example of something that was developed by this Magaloff group where we can actually add reproducibly reagents to a fixed volume of preformed plugs. So you see a preformed plug flowing along here. We have a constant flow of reagent up coming out of this fusilica capillary. And you add this fixed volume to each one based on the flow rate here and how long that droplet spins over top of that. And in this case, we're adding a reagent to initiate a color change reaction that you can see happening right there. And of course, you could use this to dilute samples as well. One thing that we were also interested in is the ability to remove samples from or to split samples as like pipette out of a sample or aliquot samples. And so Jing Ni and our group developed this little splitter that allows you to take samples that are flowing in, split off an aliquot you see going off to the right while we retain the rest of the sample. And this is actually a variable pipetter because we can vary the pressure over here by flowing oil in, and that varies the amount that we remove from the aliquot. So that allows us essentially to have a high-throughput pipetter that works on a nanoliter scale. So there is essentially this toolbox that's been developed by our group and many, many other groups for manipulating droplets. And this allows you to do a lot of different things on the droplet scale, on a nanoliter and smaller scale. And there's been many applications published, some of which are listed here. Now, as an analytical chemist, one of the things that I thought about as I looked at this field and thinking about the things we wanted to do is that we were a little bit limited, though, in terms of what we could do to analyze the contents of the droplets. We had great ways of processing them, but almost all the papers that were published were basically using fluorescence to probe what was going on in the droplet. And that's powerful, but we would certainly like to expand on that if possible. So we thought, could we do mass spectrometry analysis of droplets? Could we do separations analysis of droplets? So we've been working in this area. And I'll skip over this, but there are a number of other groups also working in these areas as well. And then after I show you how we've done this, I wanted to talk about a couple applications, especially the mass spectrometry work, for doing high-throughput screening and in vivo sensing. Two things that seem quite different, but at the end of the day become very similar types of problems. So in terms of how to do analysis, we were interested in using electrospray, mass spectrometry, and there were a number of, a couple of things have been published when we started, and after talking to Gary Velaskovic about this a little bit, we realized that maybe the very simplest thing would work. What if we just took our array of samples, segmented by oil or air gaps, and then just pumped them right through the electrospray emitter tip, keep the voltage on? Would we not be able to get electrospray for the sample when it comes out, and then either no signal or at least a very low signal for when the segmenting fluid was coming through? And indeed, this worked quite well. You can see here when a sample comes out, the electrospray starts up right away and stabilizes. I shouldn't say, it's a few milliseconds rise time there. And then when the oil or air comes out, we get no signal. The next sample comes out, again, we get a quick rise and fall there. What's happening when we have oil coming out, a lot of people are curious about that, is the oil, depending on the voltage we apply, the oil, we normally apply voltage where the sample will spray and the oil itself just dribbles off the end of the tip, and we can actually wick that away, a variety of mechanisms. And there's other things we can do if the oil does start to jet out to prevent it from entering the mass spectrometer. Either way, we've observed very little interference from the oil. Now, one of the things that we were curious about, because we do want to use this to analyze different samples through the same emitter tip, and so one of the things we were curious about is the potential for carryover. And so and what we found is that under appropriate conditions, we can really minimize that carryover. Here's an example where we made an array of alternating droplets. One contained leuenkephalin, the next one metenkephalin, and so on. And what you can see is that when you're detecting metenkephalin, there's very little signal for leuenkephalin and vice versa. And if you go in and measure the carryover, it can be quite low. It's less than 1%. And the reason for this is that the whole fluid stream is a Teflon tube, except for the very end. The oil wets that tubing, and the aqueous droplets have no way of actually mixing with each other because they're basically being carried along on this little layer of oil, and it's completely immiscible. And so that's why you get very low carryover. The little bit of carryover that does happen, we think happens right in the emitter tip, which is fused silica. We think we could actually modify that to eliminate that if we needed to. So that's how we can do mass spectrometry analysis. I'm going to come back to that later in the talk, but I also wanted to mention that we've been working hard on how to do electrophoretic analysis of droplets. And we've actually gone through several iterations with this. This is the last one that Eric Goodshaw in our group has developed. And I really like this one because it's a very simple modification of a conventional microchip electrophoresis setup. So you see here, if you're familiar with the basic CE systems have been developed by Mons and Ramsey's groups and Harrison's groups. We have a little injection cross there, electrophoresis separation channel. Here we have a sampling channel. Now, ordinarily, in a chip, you would have a little drilled hole into the chip, a reservoir there with a few microliters in it that would contain your sample, and that would be pulled up by electroosmotic flow. Our modification to that is simply to have a channel that goes to the edge of the chip and a fused silica capillary that comes out. And then we connect that to a Teflon tube here. Teflon tube contains an array of samples. When they flow by, the aqueous sample encounters this hydrophilic fused silica capillary, gets sucked in there, fills, and it's much larger volume than all this region here, fills all this up very rapidly, and then we can inject. The next droplet comes in and washes that out completely. And so the idea here, which I think is very powerful, those of you that are familiar with microchip electrophoresis, is yes, you inject picoliter volumes up there. Really cool. But guess what? You need to have a few microliters to fill this reservoir here. Now we're much able to match the sample requirements to the injection. Plus, we have a way of rapidly changing the samples on the chip. And here's a picture of this. Here's our actual glass chip. Injection cross there. These are just filled with dye so you can see them. Here's the fused silica capillary that comes out. We have our Teflon tube, and I'm going to show you this extraction here. So you see these aqueous droplets flowing down. When they encounter that fused silica capillary, they're more than happy to get out of that fluorinated environment, and they are extracted into this fused silica capillary where they fill up, and we can do injection. You see here, much like the mass spec, a sequence of injections of different droplets. They contain just a simple one-component sample that contains fluorescein at different concentrations. One volume, next concentration coming back down there. And you see the electrophoresis looks pretty reasonable there. A little bit of fronting on that peak, I think, has more to do with the injector than the fact that we extract it in this case. But the point is we have a pretty simple way now of doing electrophoretic analysis on these droplets as well. So I want to move in now and talk a little about some potential applications of this technology. One thing that we've kind of gotten interested in, it seems like a natural fit, is to high-throughput screening. So I think most of you are aware of this concept. I'll just briefly review it. But the way that drugs and chemical probes are discovered these days is to identify some target, for example, an enzymatic reaction, and then develop an assay for that target, and then take a library of compounds, hundreds of thousands of compounds, test all of them individually on that assay to see if you can modulate the reaction. And this is typically done in high-density well plates using robots to move them around. And one key feature is that the vast majority of these are done with optical readout, typically fluorescence readout. Now, this is a powerful approach, but there's no doubt there's limitations of it. The first one being that almost no biochemical reaction that we would be interested in gives you a good optical readout. Therefore, that has to be engineered into the reaction. Okay? Well, that takes skill, it takes time, and it's going to add cost to developing an assay. Once you've got it, you have to worry about false positives, because now you have another part of the reaction that any test compound we test could interfere with the indicator rather than the actual reaction that we have. A final limitation here is reagent costs. It may not be commonly known, but when you go to do a screen, they can cost $40,000 to $50,000 just in reagents, and that's the biggest expense for doing a screen. So you have to be kind of careful about when you're going to do a screen and when you're not going to do a screen. And the reason for that is both the reagents are expensive, and then when you're talking about even small volumes in a well, when you start to do hundreds of thousands of assays, it really adds up to a large amount. So what about using droplet or segmented flow mass spectrometry? What could that get us? Well, mass spec can detect the products of reaction label-free, so we don't need to build a label in. We can work in nanoliter samples, as I've already shown you, so those two things combined should greatly reduce the cost. I mean, one thing we've been thinking about is if you can reduce the volume that you need 1,000-fold, that might actually reduce the cost of a screen down to something that an individual investigator could actually set up and do very reliably rather than, without having to have a big grant just to do a screen. We already know that mass spectrometry can detect enzymatic and binding reactions, and then just as a bonus in all this, mass spectrometry is a high-resolution technique, so if you did have multiple products that you wanted to look at, or maybe you do multiplex screening, you could do that as well. So Shu-Wen Sun in our group has been investigating a couple of different approaches for doing this. The first one is something we call the mass spec plate reader, and it's essentially a two-step process. We do the reaction in a multi-well plate, then we use an automated system to withdraw a sample, move to the next well, withdraw the second sample, and so on. One of the tricks to this is to layer oil over top of that so that when you're moving the Teflon tube from well to well, you actually also pull in your segmenting fluid. And that trick was published by Viovi's group a few years ago. Once you've made your array of samples, you're then ready to pump it into the mass spec for the second step. And so I just want to show you a proof of concept that we've done with this type of experiment where we were looking at the acetylcholine esterase reaction. So this takes acetylcholine, converts it to choline. This is actually an enzyme of interest from the point of view of Alzheimer's disease and Parkinson's as well. One of the problems here is you see no chromophores, no easy way to detect these optically. So if you want to screen, what they actually do is couple this to the choline oxidase reaction, which does form hydrogen peroxide and gives you detection. But now, again, you have a second reaction that you have to worry about in terms of screening. By mass spec, we could easily detect these two guys. So here's just an example of doing that, a very small level screen, about 100 samples that we ran here. We're detecting, I'm showing you the trace, the ion trace for choline. So every time you have an inactive compound, you get this high signal for choline, and when you have inhibitors present, they're down. These are actually running triplicates, so you can see this is a better inhibitor than this one, for example. And that's a pretty, one of the advantages of the segmented flow is how rapidly you can flow sample after sample into the mass spec. So here's actually 100 samples analyzed in 2.5 minutes by mass spectrometry. So this does suggest the potential for doing screening. Now, what I've sort of challenged Shu-Wen, though, about is how do we make this truly potentially useful for high-throughput screening? What do we got to do? Well, we have to show that we can do that reformatting step. We can do it fast. We have to show that we can do long read length analysis, not just 100 samples, but start to match the size of high-density well plates. And then we need to start working on some assays that there aren't already good assays for. So move into that area. So the first thing, I was really worried about this reformatting step, that would end up being a slow step, but it turned out to be pretty easy to do. All we have to do is go parallel. Here's an example that we set up in our lab, and those of you that have been around our lab know we're not by any means expert engineers, but here's a set of 8 different 384 well plates. They're just interfaced to 8 different Teflon tubes driven by a couple of syringe pumps, and we can manipulate them all at the same time. So to show you, this is not very exciting, but each time that positioner moves down, we're creating 8 samples. And so we can create about 3,000 samples in about 6.5 minutes by that type of approach. So that's plenty fast. And they're reproducible, which was not at all a given, and Shu-Wen really had to work out the conditions to make sure this would be true. It's maybe hard to see, but if you kind of look, these are the 8 different tubes and different sections of the tube. Basically, the droplets are about the same size in the different tubes, as well as along the length of the tube. In terms of read length analysis, this is something that we've been working on, and we've been able to run pretty reliably 384 samples through one tube very rapidly. Here's an example of doing 384 individual samples in about 1.2 minutes. That's a 5 hertz analysis rate on the mass spectrometer. And you see the blow up here, how nice and reliable each one of those little spikes is that we're detecting. To do that, we have 50 nanoliter samples we're pumping through at 30 microliters per minute. So you end up with some trade-offs here. The sample has to be, when you're flowing fast to get high throughput, the sample has to be big enough to get enough scans across the peak to characterize it. And at the end of the day, we believe we're limited by the speed of the mass spectrometer that we're using. So it'd be interesting to try this on a time of flight, for instance. Okay, so the last thing I wanted to mention in this area was getting to some more interesting types of screens. And we've been thinking about, because of our collaborator, David Lombard, sirtuins, which are deacetylases. So sirtuins take a protein that's acylated, deacetylate it, they produce this acetyl ADP ribose. They use NAD and produce nicotinamide. And that reaction has been, and that can be done on a variety of different targets, but that reaction has been shown to control many very important functions in cells. So there's a lot of interest in the sirtuin family. Our collaborator recently published a paper that showed that SIRT6, which is one member of this family, is actually a tumor suppressor. And just some pretty dramatic data here. If you inject some immortalized cells that have the SIRT6 gene knocked out, they make big tumors. If you inject it with the wild type, they don't form tumors, for example, and they kind of worked out a whole mechanism here. So the idea here is we would love to have a activator of SIRT6 to be a tumor suppressor, and really anything that would modulate that gene, that protein would be of interest for better studying its function. Now, of course, with all the interest in sirtuins, there are fluorescent assays already available for looking at them, but it turns out they have a bit of a history, a complicated history. So there was a paper published that used this assay. Let me first describe the assay. This is a commercial assay. It basically takes a substrate, acylated substrate that's got a fluorophore on the end of it. It's not fluorescent in this form. Sirtuins act on it to cleave off the acyl group and the fluorophore. You can then expose it to a developer that gives you rise to a fluorescent product. And this is all proprietary, so I don't have all the exact details there. And so that screen was used for an activator of another member of this family, SIRT1. And in the screen, they identified this compound, resveratrol. You may know this as the compound that's the active ingredient, supposed to be one of the active ingredients in red wine that improves health. And they found that resveratrol might be anti-aging properties. 2 years later, 2 papers came out that said, actually, resveratrol only activates that protein when there's a fluorophore on the substrate. Okay? So this is an example of a false positive that can really lead people astray. And how they figured that out, they did that using mass spec assays. So it would really be kind of nice if we just sort of jump right to that. So we're interested not in CERT1, although we are working with CERT1 as well, but we're trying the CERT6 assay. And here's the reaction. We have a peptide substrate, and we were thinking about maybe trying the whole protein as well. NAD, CERT6, and then a deacetylate and produce these products here. So by mass spectrometry, in principle, we could look at any one of those reactants or products as a way of monitoring the progress of the reaction. We've focused in on the peptides because they're by far the easiest thing. We have the most experience detecting those by mass spectrometry. And one of the things that we've been struggling with is not really the mass spec per se, but this getting active form of this enzyme. You can't just buy it, and so we're working with our collaborator to try to get some active form, but he's given us some so far, and you can see it takes a long time to make much product. These are droplets formed from 0 time, 0.2 hour, and 8 hour, and you see you have to wait a while before you get much product form. But we get a good clean mass spectrum there. You can detect the product and the substrate. So we are making progress on having this reaction work. Now, one of the issues that we found, and those of you familiar with electrospray would realize might be an issue at times, has to do with the fact that some proteins like to work only in environments that are incompatible. So the enzymes in mass spectrum conditions, but this particular one, at least so far, we haven't found that right mixture. So what we've been doing is, if you take the reaction mixture and inject it, even if you dilute it a little bit with methanol and so on, you get kind of a very messy mass spectrum. But if you do solid phase extraction, you get a nice clean spectrum where you can see the product and substrate there. So that suggests that it's a part of this overall picture for using mass spec for high throughput screening. At times, we're going to need a high throughput way of doing solid phase extraction or some other cleanup. And there are products out there available, so that's what we're working with right now. Some of these 96-well plates that have solid phase extraction built into them, you can do them in parallel. We're also trying to work on some, Joanna Thielen in our group is working on some of our own technology that might let us do this quickly as well. But that's certainly an issue for this type of work. So that's what we're saying on that assay. Now, if I promised you when I started that one thing that we would give you label free with mass spectrometry, but also miniaturized because we're working in droplets. And then the first thing I showed you is working in multi-well plates. So that's not really miniaturized over multi-well plates. Yes, we small sample the reaction well plate. So the action assay reaction really do work at low volumes. So the concept would be that you take your library of test compounds, reformat that as a droplet, and then put them through a series of DTs where we can add different in this reaction, including quenching reagent, before we go to the mass spectrometer. And so, for example, we could add enzyme and substrate and then quench it. And I want to show you with this movie that can be done pretty rapidly, and that will not be a limiting step. I showed you a movie before. It was very slow, but we can actually add reagents to individual droplets at a pretty good clip. I think that's about 2 or 3 hertz there. The people have published up to 10 hertz, so that's not going to be a limiting factor for creating the reaction mixtures. So in this way, we would just use the nanoliter of this. We're only adding a few nanoliters to each of these guys, so we use very small volumes. The reaction we're looking at here is cathepsin. It's a protease. It's got some interest here, and again, we can just detect peptides as the products. And I just want to show you that, in fact, this works as well. We've only done this, again, on a small scale so far, but here's an example. I think about 90-some samples being injected through. When inhibitor is present, the product is very low concentration, as you can see there. And this will just emphasize that for each peak, you're actually getting a mass spectrum, so we can look at the product and the substrate. And what's kind of cool is sometimes you can also detect the test compound. So if you were worried about indexing of these droplets, at times, you may be able to detect the actual test compound right in the mass spectrum, and that gives you a good handle on what it is that you're looking at. But the real key here is not the throughput or anything, but that each reaction consumes just a femtomole of enzyme, and this is a far from optimized system. But that's really all that's used in the system. So it's a really good way to miniaturize the overall cost or the usage of samples. Okay, so that's high-throughput screening application. I want to talk about something that seems completely different at first, but as you'll see, it reverts back to something quite similar. Something we've been interested in for a long time, which is trying to do chemical measurements in the brain, dynamic measurements of chemicals. What we're interested in is measuring neurotransmitters, seeing how they change with behavior, how drugs affect the brain, and so on. And there's a lot of challenges associated with doing that. One thing I'll point out is that neurotransmitters can change very rapidly in the brain, so we like to have good temporal resolution in any measurement we make in the brain. A classic way of doing measurements in the brain is to use microdialysis sampling. So we would insert this semi-permeable membrane probe into the brain region we're interested in. We perfuse that. Molecules in the extracellular space cross the membrane, and they're transferred out where we can collect fractions and then analyze them. That would be the classic way of doing this experiment. So this is a super versatile approach because you collect fractions, and then you can analyze it by any technique to measure the neurotransmitter that you want. And so it's very commonly used for that in that regard for neuroscience and pharmacology. But it's not a high temporal resolution method because you're only flowing through these probes at maybe a microliter a minute, and if people are using HPLC or whatever, they need to collect maybe 5-10 minutes to have enough sample to analyze. Even if you have a good miniature technique, you have to worry about the fact that as a concentration pulse is flowing from the brain out to the fraction collector, it's broadening the entire time. And so you can lose temporal resolution that way as well. So this droplet approach is something that allows us to get around that. And Pong Sung in the group, when he was in our group, showed a way of doing this. Basically, we take the sample out of the brain, we immediately segment it. So that's our fraction collector. The animal literally wears the fraction collector on their head. As the droplets are being formed, we can add things like diluent, internal standards, reagents, whatever we want. We now have our array of samples stored, and they can either go directly to the mass spec or they can be stored and analyzed on the mass spec later. So we call this a mass spec sensor. It's not to drive people like David crazy who really work on real sensors, but it's just to illustrate the point that we can now use mass spectrometry to measure temporal responses in a discrete place, very much the way a sensor would do. I want to show you an example of doing this. This is going back to acetylcholine. What we're looking at is a sequence of droplets that have been measured coming from a live brain. We injected a drug called neostigmine that inhibits the breakdown of acetylcholine. And so each time we do that, we get a little burst of acetylcholine that we can detect in the brain. We know that's real change because we also added a deuterated internal standard to the droplets for acetylcholine, and it's not changing, so there's no funny business going on with the electrospray ionization efficiency here. And then again, because it's mass spectrometry, we can also look at the drug that we injected, neostigmine. So you can see here where we inject, we get a little burst of the drug. And I like to show this because it shows you the very good temporal response you get. Here's each little droplet. That's an 8-second rise time, and we're able to measure that on the system because of the good, we've used the segment of flow to prevent flow broadening in the system. And we've got the small droplets that we're analyzing. So to summarize what I've talked about today, I think these droplets let us work at what I would call a nanoliter lab. We've had a lot of techniques that can use small samples, but the preparation of the sample, the reactions, anything you need, all the sample prep, what you needed to load into the instrument, were all on a much larger scale than that. But with this droplet format, I think we can actually do the entire thing at this low volume. And that leads to the potential for low-cost and label-free screens, for example, by mass spectrometry. And that may make screening much more accessible to individual investigators. And also potential for doing things like mass spec sensing, even in a complex environment like the brain. Now, before I close, I just want to mention something about some of David's work. Again, I think we all are aware how creative he's been over the years and the different things that he's done. And one thing I thought about in this talk as I was preparing is that how, without really realizing it probably, and I never mentioned this to him, but that he was kind of inspiring to me when I was first starting out as an assistant professor, because he published this paper in Science about a fiber optic sensor. And the idea here was that he was going to use antibody-antigen reactions. And, of course, these are essentially irreversible reactions or very slow reactions. So how do you get around the fact that once you've had the binding event, you can't look at the off rate, so you can't really sense up and down changes? And he solved that problem by building into the sensor a way of releasing slowly the antibody and antigen, allowing them to react right at the tip. And then you could do this continuously and therefore get both up and down changes. Now, he did this with this clever chemistry way, and what we've been doing is using microfluidics approaches to do the same thing with antibody-antigen reactions, and sort of the evolution of that is the mass spec sensor that I showed you. So we kind of moved a long way from that initial period. But again, you never know where an idea that might look completely different down the road can inspire. And again, that's what David's creative work has done for us. So that's something I really appreciate. The other thing I really appreciate is the funding we've had to do this kind of work, as well as the students who've done the work that I described for you today. And with that, I'll close and see if there's any questions. David.
I actually have a couple questions. One is really, really incredible stuff. Can you do the droplets in the dialysis too? I mean, why not sort of flow them through and capture those rapid temporal features?
Right, right. So the even better way to do this would be to not form the droplets afterwards, but to have the preformed droplets flowing through. So we did try that, and what we found was that with all the change in direction of flow paths, the change in material they had, it was very hard to keep droplets from coalescing, from wetting the membrane, from mixing, and it just didn't work very good. That doesn't mean it can't be done, but we just haven't figured out a good way to do it. I would mention, again, is Magalov, who's worked on this. He kind of developed like an open, not a membrane, but an open sampling device that he called a chemistrode, where he preformed droplets, and he showed he could measure millisecond responses. But that was a little bit limited. You had to have like solids right next to it and so on. It wasn't really what we would want to use.
The other question I had was, it looked from your inhibitors that there were different kinetics for the different inhibitors. Can you actually get binding constants by speeding up maybe and seeing the decrease in the signal?
Right. I think for sure you could do things like vary the reaction times that you have. People have done this looked at rapid kinetics this way. So yeah, there's a lot of things that you could do in terms of looking at kinetics and binding constants. And I didn't show it here, but on the acetylcholine one, acetylcholinesterase, we did go in and do dose response curves, which are pretty easy to do that way, and so on. So you can get a lot of information, again, at the small scale. So I think it's good when, we're trying to get this good CERT6, for example. It just shows you, these aren't proteins that we buy from Sigma or something, but these are things that, like, the investigator's having a hard time making a lot of it for you. It's nice, as you do, and we're trying to do, if you can work on a small scale, literally, to start to characterize the protein pretty early on, in early stages. Hi, Rebecca.
Great stuff. As always. With the droplet-based screening, as you begin to scale it up to be truly high throughput, you're going to get to a point where you see a droplet gives you a hit, and you need a way to go back and figure out which member of your library gave you that hit. Do you have a good way of addressing those droplets?
Right. So I think there's a couple of ways of doing that. The first one I wouldn't count on, but it might work. It was what I tried to indicate when we had the mass spectrum, and I showed you that we could detect the inhibitor there. So you might have the extra peak in there, and you go, oh, that's 537. My library, that must be this, right? The other thing to remember is that when we make the way we did at least the multi-well plate system, we would know the order that we put the 384 well samples in, or 1536 samples in. So we would know this is the first droplet, and then we would have also, when we do this, and the way anyone who does screening would do is they would have both positive and negative controls put onto the wells. So not everything is an actual new sample, but it might be some kind of a test compound. So that would help you kind of keep track of where things are. Yeah. I'll be glad when that's our problem.
Any more questions? Thank you, Dr.
Yeah, thank you.